so let's talk about RSeQC. So mRNA is kind of an unstable molecule. It's kind of just made to like leave the nucleus and go to the ribosome, I guess, to make a protein. And so it's not actually made to live for a long time. And so one important thing is to check to see if your sample, if the molecules in your sample have been degraded before performing further analysis. So I think Professor Liu's example is like, if you just have like tissue stored in a freezer, you probably want to check to make sure that like the sample hasn't been degraded to the point where you can't do any further analysis. And so RSeQC is I guess it's a Python package. It's also like a collection of Python scripts that give you some measure of your sample and transcript quality. And so for problem 2, the two files that you want to focus on are this tin.py, which is going to compute a transcript integrity number, which is a measure of quality for each transcript. Then to like assess the quality of a single sample, you just take the median across all transcripts in each sample, and that gives you like a measure of the sample quality. Tin.py does this for you automatically. The second one is genebody coverage.py, which makes the gene body coverage plot. So let's take a look at an example of a gene body coverage plot. This will actually be output by genebody coverage.py. But so I guess my question to you guys is, so which samples have been degraded? I'm gonna guess why. So on the x-axis, I guess it's like different, like a list of the base pairs in a gene. And on the top, on the y-axis, is how many times each base pair was covered.
So probably all of the ones that are leaning more towards the right have been degraded.
Yeah, so like, it's probably not a good sign if like, always when you look across all genes, if like towards the 5' end, you don't see many, I guess, nucleotides in that transcript. That's probably not a good sign. That might indicate that you have like really some sort of serious degradation going on. What you want is like kind of like a flat, kind of have even coverage across all the genes, at least in the middle section of the gene. And so these are kind of what you're looking for on the gene body coverage plot. Yeah, and we'll talk a little bit more about that in a second. But so, installing RSeQC is a little bit tricky. I don't know if you guys saw my Slack post. I responded to somebody. And I suggested this as a way to install RSeQC. So basically you load Anaconda, which is like, I guess, Python and Conda, which is a package manager. And then you create a Conda environment like this. To activate that environment, you type source activate environment name. And then when you're in that environment, you can type conda install bioconda RSeQC. And this should be writing files to your home directory. And so then the relevant Python files are contained in, this is the path to find like tin.py and genebody coverage.py. So this is a little bit complicated. Has anybody tried this yet and had it work?
Yes.
So I'd recommend maybe if you have Canon free to start initiating this process. It takes like 20 minutes. Yeah, so Conda is basically like a Python package manager on Canon. It can also load at non-Python packages.
Excuse me, if we do this for the current session, will it always be installed in our Canon like home directory, or like? will we have to do it every time we want to use a Python?
Yeah, so once you do this, it's going to be there forever in this folder. But when you actually want to run it, you're going to have to do module load Anaconda and then source activate the environment. So like, I'll explain this in a little bit, but like when you go to your compute node, you're going to want to activate the environment before you run the file. And so I probably have like an instruction.
When we module load again, do we have to specify the 5.01, et cetera?
Maybe. So if you just type module load Anaconda, so there's like a default version that will load. No promises that this is the one that, like, for example, I tried it with. But it will work. Something will load if you just type module load Anaconda, followed by source activate the name of your environment. This is your choice.
Thank you.
Yeah, and so then you can actually call, like, you can type Python space path to the tin.py file. Just a standard way to run a Python file.
And does it have to be Python 2, or can it work with Python 3?
So if you type Python 2.7 here, then when you call tin.py, you want to type Python. So you'll have to check, but I installed it 2 ways, and we can talk about this maybe at the end. If this way you don't like, you can also install it some other way in which I got it to work with Python 3. But this way I would just say Python. Yeah, Python 2.
Okay, thank you.
Yeah, it's a little, this is probably the hardest thing to install on this piece set. So sorry about that, but hopefully this makes it clear. Okay, so let's just talk about how to run tin.py. Once you get it installed and you've activated your environment, basically it's, Python space path to tin.py, followed by all the options that you need to specify for tin.py. And so I've written down the relevant ones here. So -I, these are your input files. And you'll see that, so we want you to run it on like 4 different samples that are all contained in a folder. It's under raw data 2. I think it's called BAM files, might be the name of the folder. And we want you to run it on every BAM file in this folder. And so you can see if you read the documentation here, that you can just specify a path to a folder that contains 1 or more BAM files. So that's what you can do for this. I don't know what minimum coverage means exactly here. It's not super important. You can use the default value. But what is important is that you specify a reference gene model. And what I mean here is that, like, when you go to make your plot for gene, oh, this is for tin. It's the same idea though. So like tin.py will compute like a measure for each transcript. But so what you're going to tell it here, is to only use a small subset of genes. So you're not going to check degradation on like every gene that you could possibly think of. You're only going to test degradation on a select number of genes. And these are housekeeping genes. So does anybody know, like, can anybody give me like what a housekeeping gene is? Anybody heard of that?
A gene that is supposedly the expression will not vary a lot.
Yeah, so like a gene that's kind of reliably expressed under normal conditions can be called a housekeeping gene. And so it really speeds it up if you just specify that you want to, and this is good for assessing quality because, right, if you have degradation everywhere, then you probably have degradation on the housekeeping genes. So it should be sufficient just to check these. So we actually stored these housekeeping genes. They're a .bed file in the raw data 2 folder. So if you guys go there, you'll see it's called, like, it has housekeeping genes in the name. So it should be easy to find. Any questions about this?
So I guess when we run this package, we just specify refgene equals the file path of the housekeeping gene in the data folder, right?
Yep. Mm-hmm. It's a .bed file, yeah. Mm-hmm. Yeah, it has like housekeeping gene in the name.
So when we're running these commands, can we either type -i and then the path to the input file, or -- input equals the path to the input file?
Yep. Mm-hmm. So you can type -i space input file, or -- input equals.
I see. Okay.
One's just shorter.
Then the actual like full command for running this would be tin.py space Python space all of these different flags?
No, you have to put Python first. Python space path to tin.py, then all the options.
Oh, okay. And it just knows to use tin.py because you gave it the path?
Well, like the path ends with, so like after bin, there'd be a /tin.py.
I see.
Yeah.
Okay, cool. Thank you.
Yeah. Okay, so I mean, quickly, Genebody coverage, and it might have an underscore. It's the same idea. You can use default values for whatever this mRNA length is. Still, you have the same reference gene model. But you do want to call module load R before calling the script, because kind of strangely, it's a Python file, but at the end, it writes an R script and tries to run the R script because it's going to make a picture like this. So just make sure that you call module load R before doing Genebody coverage. But it's the exact same idea as tin.py. Yeah, and if the question is, what should I do for problem 2? I think 2 plots or 2 charts would be sufficient. One is like, here's the tin score, median tin score, and maybe some standard deviation for each of the 4 samples. And then also, if you can make this plot using R, the Genebody coverage plot, that'd be enough. Followed by like a few sentences of like what you've done. Because I think you're pretending that you're explaining this to like one of your collaborators. Okay.
Could you explain how to make the plots again using R?
It's going to do it for you automatically. So as long as you have module load R before you run Genebody coverage.py, at the end, you're going to get something. It's going to be a file that's a .pdf. And here you can change it to PNG or JPEG if you want. But it's actually going to make it automatically for you. So as long as you can run Genebody coverage.py and you have R loaded. If you don't have R loaded, it will say, I don't know what R is, and then it will error. But you'll still get the raw data. Does that make sense? So it's just going to do it automatically. It's going to automatically make the plot.
Okay, and the plot for the tin scores, we have to make that ourselves, right?
Yeah, so I mean, there's nothing in particular we want. I mean, a table, maybe a bar plot.
Okay.
Yeah, I think, and maybe say like, a tin score can be used to measure sample quality. And then we did that for each sample. I guess like include a few English sentences instead of just like code and figures.
Okay, thank you.
Sorry, could we go over one more time how to run the tin.py? Like, where does the path directory to the file go, like before or after writing out tin.py, and then the specifications for like input file, reference gene model, etc.
Yeah, so let me, so Python goes first. So it's just like, anytime you're running software, You generally put the software name first. So like star would go first. Here, Python goes first. When you call Python, type Python, you always want it to be followed by a Python file. And so second, after you type Python, you want to put the path, you want to put tin.py, but it's possible that tin.py lives in this folder that's like 5 subdirectories down from home. So you have to just make sure you specify the location of tin.py, because if you just type Python tin.py and you're in your home directory, it's going to say, I don't know where tin.py is, and it will throw an error. After you do those two things, then you put your input file. So -i input files. And if you read the documentation here, it says you can give the path to a folder containing 1 or more BAM files. And so we specified the location of that folder on the homework. And so that's your input file. Next, you need to specify the reference gene model. This is what genes you're going to use to actually compute the tin score. And you don't have to do anything with -c. So I think just -i and -r are needed. Yeah.
Okay,
Does
got
that,
it. Thank you so much.
if we have some time at the end, I'll actually get it out and do it. Or at least like show you where tin.py lives, because it's a little bit confusing. Great. So what's here? Okay. Okay, so something that I'm definitely probably going to do next time, or maybe at the end if we have time, as a demo. So I like to only use Canon for computation, and I really don't like working on it. And so what I like to do is every time I run something, I get the output files. I just transfer that to my local computer, to my like desktop, for example. And then I run like all my programs that are installed on my local computer. And so you want to know how to transfer files from Canon to your computer. And potentially the other direction too. In case you like write a script like on your local computer and you want to send that to Canon so you can use it on a bigger data set. So the way that you copy, and this probably only works for Mac OS and Linux users. But basically I can open a terminal on my personal computer, and then I can type SCP, this stands for secure copy, just CP is copy. And then, so I type, I guess, this is like my login.rc.fast to Harvard EDU, : the path to the file on Canon, space, followed by the destination path where I want it to go on my local computer. So have people figured out how to transfer files yet? It's kind of a tricky thing.
Actually, Philip, is like if you don't want to do SCP every time, like you can sort of install an app called FileZilla, which will like have a sort of an interface.
Oh, really?
Well, I'm not sure if I can share the screen.
So can you show this at the end, like once we have it?
Yeah,
kind of like
yeah, I can. Yeah.
And then I could also do a demonstration of this. You can do a demonstration maybe,
Sure.
or maybe Thursday, because we'll have a lot of time on Thursday. So I still want to cover salmon and RSAM. But I think this is the basic command. I can send a link in the chat, which like gives you a guide on like how to use or how to transfer files both directions. But it's you'll probably want to do this for problems 2 and 3, or just problems 2, or just problem 3, and problem 6. You might want to transfer files to your local computer. So it's good to make sure you know how to do this. Quickly, I'll give some tips for problem 3. So the text file that Genebody coverage will output, I've noticed that they're not standardized to be between 0 and 1. And so you'll see that these Genebody coverage plots, they normally go from 0 to 1. And you do that by, I guess, for each sample, you divide by the maximum number of reads or the maximum coverage at any like position on the, or any percentile on the Genebody. So just make sure you standardize your values to be between 0 and 1. Yeah. Secondly, if you want to, I guess, see a demo on how to create, like, we want it to be like a 2x2 panel figure, I guess, where each panel is the Genebody coverage plot for a single sample. This link might help you out here. And if this is a problem that people are having, I can do a demonstration probably Thursday. So yeah.
What's the difference between the plots for problem 2 and the plots for problem 3?
Yeah, there's no difference except that I think we really just want you to at least get Python running because later in the course, we're gonna have to do some probably a lot more Python coding. And so we just wanna make sure that, I guess, you know the basics of Python. So I think somebody's question I had a while back, I guess if you really don't wanna make this plot in R, you can always say like, instead of giving it, you can just say in problem 2, for example, see my plot that I made in problem 3. But since it's so easy to make in R, it just does it automatically. You might as well just include both. But no, you're right, there's no difference really, other than that one's made in R and one's made in Python.
For problem 3, what type of data structure do you recommend we get the data into? For example, I try to use a dictionary and then I try to use an ordered dictionary, but it's difficult to then call indexed values.
That's a good question. I mean, what happens? So like, I know there's some some sample names on it. What if you could you just read it in as a string and split that string up? Because like, you know that each one's 100 long.
Yeah, so that's what I did. It's just that then I stuck it into a dictionary and trying to then call elements of that in the matplot structured format isn't working. So I didn't know if there was a better, so I've split the string, I've got those things. I just don't know what to put it into that's the best structure to use. Does that make sense?
I see.
I guess Panda can work better for this situation. So a package called pandas.
Okay, I'll look that up.
Yeah.
Yeah, so maybe especially Thursday, then maybe I'll actually get it out. I think the thing with Python is, firstly, I'm mostly an R user, but there's probably like many good ways to do something. But yeah, okay, so maybe I'll make a note here that Thursday, maybe I'll like do an example with like a fake data set. So yeah. Okay.
Thank you.
Yeah. Any other questions about?
I also have a question. So for problem 3, yeah, I just mentioned I want to use like the package called pandas, but like the default version in R, it doesn't have the packages. So
I see.
Yeah,
so like
I need to.
So you can maybe so you can specify the path to where your version of Python that has all these packages lives. So I've not tried to run Python in R Markdown that much. Like earlier, one option is just to do it on some other IDE that you use for Python. And then just to copy the script, put it into R Markdown and specify eval equals false. So like, if it's convenient for you to run it within R Markdown, do so, but it's not a requirement, if that makes sense. So like if you can't get like pandas to load, then just specify eval equals false in your chunk. Yeah.
Okay. And also for the Genebody coverage in problem 2, can I just like do a screenshot and then put the figure in R Markdown?
Yeah, that should be fine.
Okay, thanks.
Sorry, when you say eval equals false, that just means the R Markdown won't run that code, right? What does that mean?
Yeah, so eval equals false means that when R Markdown, so like when it gets to that chunk of code, it will print the code, but it won't actually run it. So it's just like, that's kind of what we're looking for. We want to see your code. We want to see every like line of code that you did. But right, since it's like from a different language, and for example, like we're not going to have the files that you're like using, since you're not submitting that. It's fine just to like show us the code, but it does actually have to run within our Markdown.
Thanks.